So now is really exciting part. So we're going to start our project pitches. And so here's the way this is going to work. BrainHack Vanderbilt this year, we have 10 projects that basically you attendees are going to have the opportunity to work on throughout this weekend. The project leaders have been very hard at work to put together these projects and put together these slides to share with you all. So the project pitchers, the project leaders are each going to get 5 minutes to basically go through their project presentation and kind of explain what it's about, what you guys are going to be working on. And then starting tomorrow, you will have the opportunity to kind of talk to these project leaders and actually get started working on your projects. So without further ado, let's start the project pitches. And so if the project leaders could come, or when your slide comes up, just come up to the front of the room, introduce yourself and talk a little bit about your project.
Hi everyone. My name is Changli, and I'm a first year computer science PhD student from Nerdy Lab, supervised by Professor Katie Chang. And my research is about using foundational models to extract computer vision features for some prediction of the brain's alertness and drowsy states. And so prediction of the human's alertness or drowsy states can give us a more deep understanding of humans' brains fluctuations when the person is actually inside the scanner. However, in actual real-world fMRI data sets, the vigilance or the alertness indicator is often lacking. This time, it's the time that EEGs come into the play. In the EEG modalities, there's more prominent vigilance indicator that is something that can be calculated by the alpha theta ratio. That is the power of the alpha band divided by the power of the theta band. This indicator later integrated in an algorithm called the Vigal algorithm can compute us the vigilance fluctuations of the person inside the fMRI scan as long as they have the accompanying EEG. So our project objective is that we have the accompanied EEG and fMRI data, and we're seeking out a method to train the model to learn how to predict the vigilance states of the fMRI data alone. That is something that the model can learn from the EEG data. For the EEG data, the spectrogram can provide us a very direct visualization tool of the person's brain signals. Yeah. So what we are trying to do is to compute the EEG spectrogram and the fMRI ROI spectrograms and leverage the visual foundation models pre-trained knowledge to extract the visual features associated with spectrograms from different modalities. In this way, through inferencing, we expect the model can learn the spectrogram features of fMRI linked to alertness from the EEG spectrogram that is more direct representation of humans' alert state. Yeah. So currently, my objective of the BrainHack Vanderbilt project is can provide people some practice to integrate the foundation models, specifically the visual foundation models, for example, CLIP or SAM or Dino or other things into a deep learning pipeline and give a deeper understanding and interpretation of the fMRI and EEG resting days spectrograms. And these are the collaborators' number requirements, and any collaborators with their contribution with their method experimenting will become co-authors in following up co-publications. And for the requirements, theoretically, I would prefer some basic knowledge in programming, Python experience, or you have experience with other programming languages are preferred, and some basic knowledge of foundational models, and some basic knowledge of Git. If you have deep learning experience and would like to collaborate with us on this project, that is very welcome. The QR code of the GitHub repository and the database is linked below. And thank you so much for paying attention.
Okay. Next up, we have trial and error.
So my name is Adam.
I'm Ansley.
And we're both a part of the Wallace Lab, and we're both sensory neuroscientists. We really study behavior a lot of times. So our goal with this project is to build it's three parts. We want to build pipelines to analyze individual, condition level, and group level pipelines for analyzing what's called trial history effects. And that's kind of like a really general term. So one little like game I guess we like to play is if you look at this image right here, what do you guys see? Like someone just shouted some mess. Duck, rabbit. What if I said can you see a duck though? Can everyone see a duck? Everyone said, can you see a rabbit? Can everyone see a rabbit? That's kind of like the basis of what trial history effects are. So we can kind of what we would say prime. So what you're primed to expect kind of influences what you would consider your current perception is. And you can do this in all sorts of psychophysical tasks. And that's kind of what we want to do is build these robust pipelines. So even you, in any of your behavioral data that you collect, you're able to analyze your prior trial and trial history.
And our goal throughout this project is to develop an open source tool that can be applied across multiple different data sets. So we have 3 different data sets from our lab that we will be sharing, but hopefully other researchers could use this tool as well. And we want to figure out ways to develop visualization tools and kind of just apply this modeling approach with Bayesian modeling to this project. And we are open to anyone who's interested in learning more about behavioral neuroscience or decision making. And we'll be doing most of the coding in MATLAB, but there's also opportunities to help with like documentation and things like that.
Yeah. Sorry, elevator pitch.
Next up, we have dynamic neural decoding for adaptive neural processing.
Hey everyone, I'm Daniel. I'm a second year PhD student in biomedical engineering under new professor Daniel Gonzalez. So I'm definitely more of like a hardware guy. What our lab does is we create flexible electrodes that allow for high stability, really robust electrophysiological recordings in mice. And what this allows for is that you can have chronic recording situations for behavioral tasks where acute silicon-based probes might not be well suited. So over the course of the last year, we've been making these probes, we've implanted them in mice, and now we have recordings. We are generating, and able to identify spikes from visual cortex of these mice. And really what we now want to do is bridge that with some form of a decoder to kind of have a behavioral effect that we want to find. And that's really found in the adaptive learning behavior. So you can imagine if we were to put this probe in the mouse's brain and live record over the course of days, weeks, months, as we train the mouse on some sort of basic, what we call a cursor task. Let's select 2 populations of neurons along the length, the shank of this probe that are responsible for controlling maybe in the simplest form, the direction of a cursor 2 ways. And you can provide some sensory feedback that if you're driving the cursor left and you reach a target, you're rewarded. And as the mouse becomes accustomed to this, you've conditioned it. But what we're really interested in is if in the middle of this learning period, can you switch the subpopulations of neurons that are being, driving one direction or the other? And you could also think about, multi-directional, how this relates to the timing of the movement of the cursor. There are many ways you could alter this. You then want to study the neural effects of these changed subpopulations while the mouse is trying to adapt to this behavior. So really, at the core of what we want to do in BrainHack is to create a decoder. Simply, we have our own recordings from the lab, we have some sample recordings from different labs, and create some form of a visual cursor that will be driven by subpopulations of electrophysiological recordings. So we're open really to anyone with any amount of skill set, MATLAB, Python, whatever your background is, or if you just want to learn more about reading brains, reading minds, I guess. But yeah. Ask me questions. I'm excited for the weekend.
And next up, we have an all-optical brain-computer interface.
Click on the mouse pad to go to the next slide.
Yeah.
Okay. Technical difficulties.
Okay, there.
Hi everyone, I'm Amelia, I'm a first year PhD student in the Gonzalez Lab.
And I'm Max, I'm a fourth year undergraduate student.
And we're working on all optical brain-computer interface. Specifically, like Daniel mentioned, paradigm in the Gonzalez Lab works to integrate cutting-edge neuroscience engineering to understand the brain and how it functions on 3 levels, specifically at the subcellular level, the circuit level, and then the systems level. This project is specifically focusing on that subcellular level, and our goal, in comparison to Daniel's project, we're going to be using an all-optical 2-photon PCI approach compared to electrophysiology. And our goal is to develop a real-time, rapid neural decoding algorithm to control this 2D cursor, as Daniel explained, based on Gaussian transient dynamics, based on our 2-photon imaging in healthy animals, with plans to then extend this framework that we developed to disease models such as autism spectrum disorders. We're looking for people with experience in MATLAB or Python, but generally most things that you're excited in neuroscience, computational biology, and engineering. And we're hoping that you'll learn how to do motion correction, image segmentation, and fluorescence trace extractions from 2-photon imaging data, as well as be able to develop novel neural decoding algorithms that could be applied across a variety of different kind of techniques for imaging purposes.
Look it, I think.
Yeah.
So our anticipated workflow is going to consist of 4 stages. First, we're going to start with pre-processing of the raw 2-photon fluorescence images, consisting of a filtering stage for usually normalization stage, which is important for the neural network training, and outlier removal, removing false positive fluorescent signals, either using an established application like Sweet2P or Common, or using a deconvolution method. The second stage of our workflow is going to be image registration. First, we're going to tessellate the image, split it up into multiple different subregions for analysis to enable cluster analysis, and then we're going to design our transformation model, the transfer function between our original images and the motion corrected images. And then finally, we are going to implement the motion correction. This is probably the primary goal, or one of the primary goals of our project. Third phase is going to be image segmentation. It's going to consist of feature mapping. We're going to identify relevant features such as average fluorescent intensity or fluorescent variance over a short period of time. And then secondly, we're going to localize the signals and kind of identify ROIs for each of the neurons. Lastly, we're going to implement a neural network to analyze the data with all of our features. We're going to design it using TensorFlow with Python. Everything else is going to be done in MATLAB for the most part. Then we're going to train the model using our raw data frames. And finally, we're going to use the outputs of the model to determine our spike inferences. Yeah. Thank you.
Okay, next up, we have brain state modeling.
Speak in front of the computer.
All right, hi everyone, I'm Bruno. I'm a first year PhD student in the BN lab. We are housed in the Vanderbilt neurosurgery department, and we research epilepsy, trying to understand it better and develop better treatments for it. We get our data mainly from epilepsy patients that fail to respond to medications, and so they come to the hospital to get worked up for potential surgical treatment. This typically involves placing electrodes in their brain. This can be up to 100 electrodes, and then they wait in the hospital, and we collect data, which really gives us a privileged opportunity to get intracranial EEG data from waking humans. Unfortunately, this data can be pretty hard to interpret, both from a clinical and research perspective. And so our lab has been working on this deep learning model that essentially tries to represent this multi-channel time series data as brain states. And so the details of the model, I'll forgo for the sake of time, but the general goal is to basically feed in this multi-channel time series data in 10-second chunks, and for it to kind of summarize these dependencies between the channels as a single high-dimensional vector that kind of captures the global activity of the brain, and that's what we call a brain state. By itself, that doesn't really mean much, but what happens when we feed in those 10-second chunks across the patient's entire hospital stay is that we get this high-dimensional point cloud that essentially captures the variety of functional brain states that the patient occupied during their stay. And so you can imagine that the patient kind of wanders around this point cloud as they're awake. When they start seizing, we see that they kind of go to a specific part of the point cloud. And there's a lot of directions that we can take this. What we're most excited for is potentially using this in a closed loop neuromodulation system where we can detect brain states that are of high seizure risk and then neuromodulate away from those brain states towards safer brain states. But really, that involves picking up on nuanced functional states that is kind of a downstream application. And what we first wanted to do was validate this model's ability to actually differentiate between functional brain states. And so luckily, the human brain kind of cycles through a set of very unique functional brain states, which are sleep stages. And so we've been using that to validate this model. We took data that we had about when the patient was awake, when they were in NREM, and when they were in REM sleep, and we labeled this point cloud. And as you can see, it seems like the model is able to pick up on differences between these functional brain states. But ultimately, this is still depending on external data for labeling. And so really what we're hoping to get out of this weekend is to actually develop some kind of way of differentiating or classifying sleep stages purely based on this point cloud and these embeddings. And so that might take the form of a classifier or a decoder. Really, we're open to ideas, but the idea is to kind of further flesh out the ability to detect brain state or sleep stages using this model. Other kind of projects that we'd like to get out of this is just experiment with other manifold projection techniques. We've been using PackMap, but that's just kind of something that we chose up front. And, even just pipeline development, it would be, it'd go a long way in terms of it helping us be more productive. So we're really happy for any directions, and I'm happy to answer any more questions if anybody wants to talk about this. Thank you.
Next, we have less confusion in diffusion. Speaking finally.
Okay. Hello, everybody. My name is Nancy Newland. I'm a fourth-year PhD student in the Massey Lab, and my main focus has been in diffusion-weighted imaging. And this is my co-leader, Elias Levy, and he is a research technician also in the Massey Lab. His experience is in large language models. So in this project, we are bringing together both of our expertise to bring you less confusion in diffusion. So the main goal of this project is to create an image to text bot that will give you suggestions for your diffusion weighted image pre-processing. The overall goal for this weekend is to create the basic pipeline. Briefly, technical details include using a pre-trained vision language model and being able to take user input of a screenshot of your diffusion image and provide you with a list of what's wrong with the image and hopefully a cure or a list of solutions for that. I'm really excited to learn about the language model part of that. And if you are too, then you're more than welcome to join us. We are looking for people with like intermediate machine learning experience, comfortable with Python, and eagerness to learn about medical imaging and the language model part. I think that's it. Yep, looking forward to it.
Okay. Next, we have OpenDive, Open Diffusion Imaging Visualization.
Hello, my name's Adam Saunders. I'm a second-year PhD student from the Massey Lab.
I'm Alyssa McMaster. I'm a second-year PhD student also from the Massey Lab.
And our project is open diffusion visualization for everyone. So we've been talking about diffusion imaging tonight. It's very common for us to encode the idea of direction in our visualizations with this red, green, blue color mapping. Unfortunately, about 5 to 10% of men and about 0.1% of women are red-green colorblind. And so one of the goals of our project is going to be coming up with a color mapping that is accessible to people with color blindness for these models. And Alyssa's going to talk about our tractography aim.
Yeah, so we've already had a fabulous introduction to tractography tonight. So now that you all are used to kind of seeing this kind of image, we are really interested in using the same colorblind friendly maps that Adam just mentioned, but we're also interested in using these different diffusion bundles to visualize changes over time that happen in the brain, maybe during different diseases or disorders, or maybe during the aging process. So we're trying to look for a way that we can summarize those changes and visualize them in a nicely wrapped up Python package.
Yeah, we're looking for people with a little bit of Python experience, but if you've never worked with this imaging modality, that's great. We'd love to have you. And yeah, all right. Thank you.
Thank you.
Now we have deep MRI.
Hi, I'm Elton Cross from the Land Lab, and I'm working with my fellow project leads, Ashley Howe, Zoha Arif, and Dr. Vinci Boer, to use a neuroimaging tool called Deep MRI to estimate eye gaze patterns from fMRI data. So essentially, we're using deep MRI analysis of eyeball voxels extracted from fMRI data to estimate gaze patterns during the scan and validating that output through comparison with actual in-bore eye tracking measurements. I'll explain this a little bit. So normally, if we want eye tracking data during an fMRI scan, we obtain it by using a hardware system that measures light reflecting off the pupils, like I have up there. But this kind of in-bore eye tracking is not always available, and deep MRI can help us evaluate eye gaze when this traditional method is unavailable, because it uses changes in the fMRI signals of the eyeball voxels to infer where those eyeball voxels are looking at, as in the second picture right there. It's very important to note that deep MRI does not require a special MRI sequence either, meaning that it can be applied to any existing fMRI data set, including the large open neuroscience data sets, like the ones that are available for us all to use. So our overarching goal here is to investigate whether the measurements of eye gaze patterns calculated through deep MRI align with the measurements of traditional in-bore eye tracking. And if this is the case, then deep MRI may serve as an alternative method to obtaining eye tracking data, while also giving us insight into how the neural correlates and the neural correlates of attention and visual processing are modulated by gaze behavior in data sets where this would have been previously impossible because they lack that eye tracking data. So we are welcoming anyone to participate with us in this project, regardless of skill level. However, we want to make sure that at least one team member has experience in neuroimaging tools like FSL, one team member that has experience using Python, and another that has experience with version control such as Git and GitHub. You will learn a lot with this project, namely how to use deep MRI to infer gaze patterns from fMRI data and develop visualizations to validate deep MRI outputs against traditional eye tracking data. And after replicating the original deep MRI results using their practice data and documentation, project participants will apply deep MRI to pre-processed fMRI data that we acquired here at Vanderbilt. And we will be starting with an adult data, and then we will move on to trying it with children. So importantly, it's important to note that we have concurrent in-bore eye tracking data for all of our fMRI data set that we plan to use. And this allows us to validate the deep MRI measurements against standard eye tracking measurements. More specifically, if you participate, you will be downloading the weights for the pre-trained deep MRI model and using the provided cross-validated code in Python to extract the eyeball voxels and estimate the gaze patterns of our fMRI data set. Any contributors will be acknowledged in our GitHub repository, as well as cited in any related publications, and may have the opportunity to continue collaborating on future projects.
And that's it. That's our project. And together we can help validate an alternative method for obtaining eye tracking data in fMRI scanning and advance our collective knowledge of neural correlates and gaze behavior. You'll become experienced in using both eye tracking and fMRI data. It's very exciting. I hope that you guys are interested in joining us, and we're looking forward to working with you. Thank you.
And now we have nonverbal IQ modulate the relation between physiological awareness and reading skill, harmonizing multi-site MRI.
That was a little intense.
Yeah.
There you go.
Hi everyone. My name is Isaac, and I'm a first-year master's student in the cognitive psychology program.
Oh, sorry. Is there like a, gotcha. No problem. So I'm a first-year master's student in the cognitive psychology program here at Peabody, and I worked in the brain development lab with Dr. James Booth. And so at our lab, we are interested in understanding the development of reading and language and explore how this process is connected to other cognitive functions. And so one of the questions that we are interested to learn more about is whether the neural basis of reading depends on IQ or not. And the way we're going to tackle this question is by leveraging hundreds of fMRI data that our lab has collected over the years using variations of a task that has been at different locations using different scanners. And the advantage to doing that is it not only boosts our statistical power to detect changes and all that, but it also makes our findings more generalizable to a diverse population. But when combining multiple neuroimaging data sets, there comes with a major drawback, and that is it introduces non-biological variability in our data. And this can lead to misleading results if it's not addressed. And so that leads us to our project here at BrainHack Vanderbilt. So our goal is to develop a harmonization pipeline to remove between scanner variability and to ensure that we have reproducibility and trust in our later downstream analyses. And so the tool that we hope to use for this harmonization is called Combat. And Combat is a tool that originated in genomics to remove between lab variability in terms of how they process their cell samples. And this tool has since been adapted to neuroimaging data, where it has proven to be effective in harmonizing MRI data sets across multiple modalities. And Combat is also widely used in large-scale studies such as the Alzheimer's disease neuroimaging initiative, ADNI in adults, as well as the adolescent brain cognitive development study, ABCD, in children. And so for this project, we are looking for people who are comfortable with using R and MATLAB. And to give you guys a quick snapshot of what we hope to do over the weekend, first, we will do a quick tutorial for everyone to get you feeling comfortable with using Combat. And then we will use Combat and apply that to a sample of data that I've selected from our 3 open neuro data sets. And then afterwards, we are going to try to assess the effectiveness of harmonization on our data and try to understand how motion affects the harmonization process. But don't worry if you are not a strong coder or if coding is not your strong suit. I believe we also have a lot of other opportunities available for anyone who wants to contribute to our project. For example, reviewing more literature about harmonization, helping with documentation of our pipeline, and also like brainstorming ways for us to visualize the effectiveness of harmonization. And if time permits, we would also love to explore more newer advanced techniques for harmonization, such as machine learning approaches and deep learning techniques. So if you're interested in joining this project, I think you'll gain valuable experience with processing fMRI data. You will gain insight into identifying and addressing variability for multi-site fMRI data, and learn about quality control. And if you're an expert coder or if you're someone who just wants to get more involved with neuroimaging research,
there is a place for you in this project. And so if you're interested, please feel free to reach out to me and we can talk about more about that. Other than that, thank you everyone for listening, and I wish everyone a great brain hack.
Thank you so much, everyone. Fantastic projects. I'm excited for all of them. You guys are going to see me hopping around, hacking with you throughout the weekend. We are closing the project pitches. We have still plenty of food. So if you guys want to mingle and talk about the projects, you're welcome to do it while you're, taking advantage of the great food we have. But we do have a structured project shopping period tomorrow from 9 a.m. to 10 a.m. Okay, 10 a.m. to 11 a.m. We will basically give each project leader a table, and we have a really cool plan of doing something very slow paced, speed dating mixed with musical chairs. It's gonna be our kind of like theme for doing the project shopping period, meaning project leaders will explain more in depth their project to you, and if you're interested, you're gonna get to talk to them more. So thank you all so much for coming. I'm really excited for this weekend, and yeah, let's do it.